Dear students, Assalamu alaikum. Today I am going to deliver a lesson for class 12th, subject zoology. My today's topic is the process of recombinant DNA technology. The recombinant DNA technology is a very complex process and involves many steps to achieve the desired objective. The major steps involved in recombinant DNA technology are: number first, the isolation of genetic material, that is DNA. Since DNA is enclosed within the membranes, therefore it is necessary to break the cell with the help of enzymes to release DNA. Before cutting the DNA with restriction enzymes, it needs to be in pure form, that is free from other macromolecules like RNA, proteins, polysaccharides and lipids. Now the methods used for the isolation of genetic material, that is DNA, is shown in this sketch. First of all, we get a living cell or a tissue from which the DNA is obtained. Since this DNA is enclosed within the cell membrane, so first of all we have to break these membranes by using various enzymes like lysozyme, cellulose and chitinase, depending upon whether animal cells, plant cells or fungal cells are used for obtaining DNA. After breaking the cell membranes, we get a pre-DNA with other macromolecules like RNA, proteins, polysaccharides, lipids, etc. Now in this next stage, we remove RNA by treating this suspension with another enzyme called ribonuclease that destroys the RNA, and we get a DNA with proteins, polysaccharides and lipids. Now from this suspension, we remove proteins by treating this suspension with another enzyme called protease that removes the proteins. Thus, we get a suspension which contains only DNA with polysaccharides and lipids. And in final step, we treat this suspension with chilled ethanol that removes polysaccharides and lipids, and in this way we get a purified DNA in the form of thread suspension. After this stage, the next stage is the cutting of DNA at specific locations. Now under the optimal conditions, the purified DNA both from the vector and target DNA fragments are separately digested with the same restriction enzyme. The digested vector and the target DNA fragments are incubated together in the presence of DNA ligase. This results in joining the two types of DNA by formation of phosphodiester bonds between them, forming a recombinant DNA. After the second stage, there is a very important third stage called the amplification of the gene of interest using PCR, that stands for polymerase chain reaction. This PCR polymerase chain reaction is basically DNA replication in vitro. The PCR was invented by Carey Mullis in 1985. The PCR results in selective amplification of the specific region of a DNA molecule and therefore can also be used to generate a DNA fragment for cloning. The basic requirements of a PCR reaction are: number first, a DNA template, 2 nucleotide primers and enzymes, usually DNA polymerase. This DNA polymerase is also called Taq polymerase. It is a thermostable enzyme and can withstand the temperature of more than 90 degrees Celsius. Now, a single PCR amplification cycle involves three basic steps. These three basic steps are denaturation, annealing, and polymerization. So far this denaturation is concerned, in this step the target DNA is heated to a high temperature, usually 94 to 96 degrees Celsius, resulting in the separation of the two strands. Each single strand of the target DNA then acts as a template for DNA synthesis. Second step is the annealing. In this step, the two oligonucleotide primers called anneals are hybridized to each of the single-stranded template DNA, since the sequence of the primers is complementary to 3' ends of the template DNA. This step is carried out at a lower temperature, usually 40 to 60 degrees Celsius, depending on the length of the sequence of the primers. The final step is the polymerization, also known as extension. The final step is extension, where tag DNA polymerase of a thermophilic bacterium called Thermus aquaticus synthesize the DNA region between the primers. It means the primers are extended towards each other so that the DNA segment lying between the two primers is copied. The optimum temperature for polymerization step is about 72 degrees Celsius. Now, to begin the second cycle, the DNA is again heated to convert all the newly synthesized DNA into single strands, each of which can now serve as a template for synthesis of more new DNA. Thus, the extension product of one cycle can serve as a template for subsequent cycles, and each cycle essentially doubles the amount of DNA from the previous cycle. As a result, from a single template molecule, it is possible to generate 1 billion molecules after 30 cycles. Now, this is about the isolation, cutting up DNA, and the amplification of the gene called PCR. In our next topic, we will discuss about the other stages of the recombinant DNA technology. Thank you.